Hi, hello everyone. Welcome to today's Cell-Press Forum on Sustainability. Today's forum, Land Health, Preventing Zoonosis Spillover, will focus on themes of land use changes, zoonosis emergence, ecology and transmission, biodiversity dynamics, and interdisciplinary approaches to disease management. My name is Ryan Perry. I'm an editor at iScience, Cell-Press's journal for interdisciplinary science. Along with my colleagues, Carrie Tate and Abigail Kelly from Cell-Press's flagship sustainability journal, One Earth, we're excited to host this discussion with our keynote and panelists and answer your questions from the audience. I'd also like to thank Sarah Rooker and Lauren Manges at Cell-Press, who've been instrumental at producing this event. Before we begin, I wanted to highlight that many of the journals at Cell-Press publish research related to the topics covered in today's forum. Editors at One Earth, iScience, Current Biology, Patterns, Trends in Microbiology, Trends in Parasitology, and Trends in Ecology and Evolution have created a collection of recent articles from across the Cell-Press portfolio. It's available if you click the right side of your screen, Handouts tab. That collection is available as well as a few other collections from Cell-Press. We hope you enjoy them. Just quickly, in terms of format, following this brief introduction, I'll invite our keynote speaker, Chris Murray, to give his presentation. And then we'll invite our panelists to speak about their research areas. Following that, we will have a moderated panel discussion and then wrap up with a few closing remarks. Before we get started with the talks, we have a few practical points. If you have a question for any of the speakers, you can use the Q&A box on the right of your screen. You can enter a question at any time or upvote questions you would like to be answered. We'll try our best to pass on as many of the questions to the speakers as we can during the Q&A, time permitted. It's also helpful if you have a question for a specific speaker that you include their name in your question, so we'll know who to call out for the response. And now, without further ado, I'd like to introduce today's keynote speaker, Professor Chris Murray. He's the co-director of the Center on Climate Change and Planetary Health at the London School of Hygiene and Tropical Medicine. He is an ecologist with interest in global environmental change and its linkages to human, animal, and ecosystem health. He investigates the relationships between environmental and social influences on health, focusing on infectious as well as non-communicable disease outcomes, disease vectors, and zoonotic hosts. So, Dr. Murray, the floor is yours.
Thank you, Ryan. Thanks so much, and thanks to the organisers for firstly organising and secondly inviting me to give a very inequitable keynote here. The panel is absolutely top shelf today, so I'm really pleased to be part of this. As per the introduction, my name is Chris. I'm based at the MRC unit, the Medical Research Council unit, the Gambia, part of London School of Hygiene and Tropical Medicine. And here I lead out our work on planetary health. I'm going to kind of keep my introduction as sort of general as possible because I know that the topic experts that we have today are going to dig in a lot on some of the content that we're covering. So I hope it's a general introduction and stimulation for conversation. So I want to start with the sort of high-level context of this topic, and that's really about, at least in my mind, around anthropogenic global change. And the global change that we've seen in recent decades is really of a scale that's sort of unprecedented. It's relatively recent, and it's extremely variable, heterogeneous, spatially and temporally all over the planet. Things are happening in different ways and in different places. But the big trends include things like huge change in climate, more than a degree of average warming since the pre-industrial revolution, and that's increasing at about 0.2 degrees per decade. We've also substantially modified more than 50% of the global land surface. There's been a threefold increase in global population just since 1950 and a 56-fold increase in international travel since 1950. And these things are culminating and changing ecosystems and ecology the world over. One of the most dramatic effects of these changes is this huge impact on biodiversity, and we're in a situation now with around about a third of all species, that have been assessed are at risk of extinction, and this is relative to a background extinction rate. The extinction rate has probably increased by 100 or maybe even 1,000 times. Land use change is one of the major drivers of these shifts, as you can see for the figure on the right. It's really a driving force of biodiversity loss, and in addition to that, we have direct exploitation of biodiversity, as well as climate change starting to have an effect as well. So this is the really sort of high-level environmental context of what I'm going to talk about today. If we move to human infections, the story is actually kind of the opposite, and I think it's pretty clear from this burden of disease diagram to sort of say that, we've probably never been better off in terms of a species, the human species, in terms of our health, and all the metrics of human health on average are going in the right direction. Life expectancy is increasing, mortality from infections is decreasing. But that's not the full story. And, if we consider endemic infections, there's still a couple of hundred zoonotic infections and a couple of hundred vector-borne diseases that are causing billions of illnesses each year and millions of deaths. And in addition to that, it's not all a pretty picture when it comes to disease control. And we have certain diseases that sort of buck the trend, so to speak. And SARS-CoV-2, cause of coronavirus, or the pandemic coronavirus that we've all lived through now, and everybody is a personal expert on, really demonstrates how quickly a new infection can shift the global landscape of the burden of disease. And that uptick that you can see in that final couple of years of this chart is an example of an emerging infection. Emerging infections are, common. These are emerging and re-emerging pretty regularly. There's at least a few per year, new diseases that pop up on the radar. They're often challenging, but occasionally, I would say maybe very occasionally, they become catastrophic. Examples of the pandemic that I just mentioned. For the most part, emerging infections are coming from animals, so they're zoonotic, and for the most part of those, they're also coming specifically from wildlife. About almost half of emerging infectious diseases are coming from wildlife specifically. And there's been a lot of work, I'm not quite sure what happened to my image there, but there's been a lot of work trying to work out whether the frequency and the magnitude of emerging infections is on the increase. And I think the sort of consensus is that it probably is, although I think we do need to recognize that there's some very serious data challenges and analysis challenges when it comes to trying to really pin down this trend. This is a figure that I pulled from a pretty recent publication that shows some pathways or at least studies that have characterized pathways and linkages between climate change, biodiversity, infections. And you can see the literature is coming, really from literature reviews, but also theoretical studies and empirical studies, like field studies, covering a whole range of different ecosystems, a whole range of different taxa. The thing that I want to sort of show with this figure is just how diverse the mechanisms that have been identified that link these factors really are. It's a very, very diverse and complex space when we're considering the processes of how climate and biodiversity influence infectious diseases. I sometimes use this very simple schematic model. I call it an infection engine, so to speak, where I think that there's this sort of like core features of anthropogenic change that is driving or at least influencing the ecology of infections. Land use change on the one hand, biodiversity loss and change on another, and on a third hand, climate change as well. And these things are not independent, they're working all together, and they're sitting in the context of massive human ecological change, changes in mobility, changes in socioeconomics. And I use this diagram on the right sometimes to sort of illustrate the place-based nature of infections, that the influences are often coming from the places, the geography of those places, the environment of those places, the species of vectors and animal hosts that are in those places, and the pathogens and parasites that they can support, the people, of course, that live in those landscapes. And what we know about diseases, we sort of see through this sort of fuzzy lens of research that is influenced by a whole bunch of different things. How good we are at reaching different parts of the globe to observe, and things like, this constant effort to manage disease and stamp them out in some places. And it's the culmination of all of these factors that give rise to landscapes that are either more or less disease prone. And you can sort of look up this diagram from the bottom and sort of appreciate this on a single disease type, from a single disease type perspective, but you can also look down as if you're a bird or a satellite and start to see patterns that emerge at a global scale around the sort of the patterns that structure whole infectious disease communities, over very, very large spatial scales. So coming to the topic of today's webinar, this idea of what people have frequently referred to as unhealthy or pathogenic landscapes. I myself prefer the term diseasey landscapes. I just find that to be somehow funny for not a very funny topic. But this is really the idea that there's stuff that we're doing in landscapes in particular, particularly around land use change, that's altering the ecology of disease and causing unexpected disease problems. And I've pulled a couple of titles from a bunch of papers that were very influential to me when I was sort of wading into this field. And it's really great that I think everybody that's on this panel today has contributed to this single diagram that I've put there on the right hand side. So it's fantastic to be sharing a platform, with practitioners in this field and influential practitioners in this field. This is another result from a systematic review, a really large review looking at the linkages between climate change and infectious diseases, done by Mora et al. A couple of years ago. One of the categories in that study, climate factor or climate hazard, was land use and land use change. And you can see from these wiggly grey lines, just again how diverse and how diverse and how many different pathways have been observed in empirical and other studies that link the change in land cover and land use, and all of the many different mechanisms in that, all of the many different types of transmission pathways, and all of the many different types of diseases and pathogens that are involved. I really like this study because you can actually go into this database and you can click on those grey lines and it returns the study, so you can really have a great immersive experience in a sojourn in disease ecology. So coming back to that diagram that I mentioned, this sort of conceptual model, the real question to me is not so much whether we can observe individual cases of land use and land use change affecting or creating novel disease risks for people, but I think the bigger question is, are those landscapes in themselves inherently more diseasey than they would have otherwise been? And I've sort of tried to represent this process of, if there's some kind of change that is culminating in the change of the environment and the change in the biodiversity that supports disease, which is, again, the diversity of pathogens, also another form of biodiversity, on average, is what we're doing to landscapes, is that making that landscape fundamentally more pathogenic, more diseasey, more unhealthy, or is it doing, and how is it doing that? Is it removing the services that nature provides in keeping a lid on some of those infections? Is it selecting for better disease or more disease hosts or vectors? The habitat preferences of all of these different species, and again, this very complex ecological space, what's the sort of net effect of all of that? There are plenty of suggestions around this, and this is one that I like from Raina Plowright and her group that's sort of been gaining widespread use over the last few years, this idea of land use induced spillover, and connected to this idea of landscape immunity, the idea that these impacts that we're having on landscapes is fundamentally changing the sort of pillars on which disease ecology is based. If we go to empirical research that tries to sort of test this idea, this is a study that a student of mine did a few years ago, looking at whether people in agricultural landscapes or people that are exposed to agricultural landscapes, so they're landscapes that are very heavily modified typically for growing food, are those communities inherently at more risk than other communities for infections. And if you control for all of the different things that you might think might confound that type of relationship, you end up with something like this, where it does appear that people in these impacted landscapes are around about twice at risk than the general population, or people that are not exposed. Here are another couple of studies that I think help illustrate this kind of idea that there could be something to this net impact, this net effect. On the left, we have a study by Rory Gibb and team looking at the change of the proportion and the abundance of zoonotic hosts, so hosts that we know are carrying around pathogens that can infect people. And what they found is if you gradually change the landscape, or you move through categories of landscape from very natural through to very modified, the proportion and the abundance of those zoonotic host species appears to increase. So that represents this sort of natural baseline of risk for people in that landscape contracting these diseases. Of course, there are many things that push back on that. So landscapes that have more people might have more infrastructure, might have more resources, might have better connectivity, might have different ways of combating infection. So the net sort of impact on humans in terms of the infectious disease burden may not necessarily be higher in these landscapes, but it's that underlying basis that is of interest here. And on the right-hand side, we have a summary of a lot of different studies collated, again, in a review and a meta-analysis that showed that the basis of this change in ecology, this increase in diseasiness, is probably coming from the removal of biodiversity, which is in some way buffering or diluting, so to speak, the sort of the infection risk to some target host, and in our case, that would be we'd be considering ourselves. So I wanted to just give a very quick sort of example of how sort of I see this kind of playing out. I'm going to take the example of food systems. Food systems are clearly very important, very useful. We need food systems to improve our nutrition and grow and keep healthy and all the rest of it. Food systems are also a good way to help communities develop in some way. It leads to development of infrastructure, roads, schools, hospitals, and livelihoods and things like that. But and at the same time, it is getting more efficient, the food system, but it comes with a lot of very serious costs. And the one that I mentioned initially was this major driver of land use and land use change. Also a huge contributor to greenhouse gas emissions and climate change. And at the same time, it's not really fulfilling the function that we need it to fulfill, in the sense that we still have this big gap in basic nutrition. Around about 1 in every 10 people remain undernourished at a global level. Around about a quarter of people remain moderately or severely nutritionally insecure. Around about a fifth of adult deaths are associated with the poor diets that we're increasingly eating. And there's been this huge reduction in diversity of food sources, around about a 75% reduction in crop diversity since 1900. And now 17 crops, 12 food crops and 5 animal species, or I should say, provide around about 75% of the world's food energy supply. We also know that reduced food biodiversity can also have direct impacts on human health, as illustrated by this study that I was involved in, looking at cancer risk in a European cohort. The other thing that I think is really important about food systems is it's an illustration of where you've got global demands that are connected to very, very local impacts, what I think is being referred to frequently as teleconnected, and something that I know Professor Eric Lambin has pioneered is this sort of connectivity between consumers and producers, and thinking very much about the impacts that those drivers have. And there's been some examples linking that process to disease. The top figure here is looking at the malaria risk, the increase in malaria risk that is driven by commodity in forest products, so deforestation. We did have a little crack in my group at trying to do that for the global burden of disease associated with agriculture, based on that review that I mentioned earlier. So looking at sort of the sources, the drivers, and the sort of the drivers and the receivers of these infectious disease impacts linked to trade for food systems. But that really prompts the question, what does a healthy food system even look like? I'm not sure that there's any universal answer for that. There's been many suggestions of, the types of things that go into sustainable food systems. What does it really look like, and how do we know that what we're doing in landscapes, particularly as it's relating to agriculture and things like that, is not causing more problems than it's solving? And I think some really interesting questions here. The gradients of intensification, this kind of idea around sort of optimizing land sparing and land sharing. We need some way of measuring the value of alternatives when it comes to shaping our landscapes, particularly for food, and understanding this idea of whether this has ultimately got some potential. The mechanism, the intervention has some kind of potential to reduce spillover risk. Is a really important question. And then just to leave this with just some closing thoughts, there is a lot that's already happening globally to sort of prevent and reverse the degradation that I've been talking about, the things that we know are connected to infectious disease risks. There's huge numbers of international agreements to try to stop doing the things that we know are particularly harmful, including greenhouse gas emissions and deforestation and things like that. Many of these interventions will have implications for health, and yet my observation is that health is not really at the heart of most of this discussion. And the evidence around these change, the response, the degradation and the response, health practitioners and health stakeholders are often missing. I break this up into sort of two very broad, rough categories. That is preventing this degradation, preventing this land use change on the one hand, and then restoring on the other. Most of the literature that we've got around land use and infectious disease emergence and risk is on the side of degradation. So it's on the side of impacts. But what are the benefits of preventing and restoring? Does the directionality of this process matter? There's lots of reasons to think that it does matter. There's lots of studies to sort of show, as I mentioned, this process, and there's reasons to think the prevention of further degradation will keep a lid on these processes that catalyze disease risk, things like preventing biodiversity loss and leading to the prevention of dilution effect loss, preserving landscape immunity, as I was referring to earlier, preventing land use induced spillover, and preventing the spillover and onwards sort of escalation into pandemics. On the restoration side, I think it's a lot less clear. It's really a lot less clear what the implications of many of the really big fields that are emerging in response to sort of thinking about this sustainable transition, things like restoration, climate change adaptation, nature-based solutions, regenerative agriculture. We really don't have many studies that incorporate the health dimension there. Some of them are infectious disease related, but very few of them draw them all together. And this is a point that one of our panelists today has made in this recent paper, and I fully expect that this will get a little bit more coverage in the rest of the webinar. So that's my sort of big picture take on some of this stuff. I'll leave it with this diagram of some kind of social ecological systems type approach, where I feel there's a lot of potential to be able to lean on sort of land system science and social ecological system science to make progress in this area that we currently haven't really made to the extent where we're sort of influencing day-to-day land use planning and land use policy. I think there is a framework for that. I'm not necessarily sure that the infection and emergence risks are the only, or even perhaps the most important health impacts of this process, but it's very important that we bring it into the frame and try to synthesize it with other health impacts and other health impacts in other sectors as well. And I think that's all I had for my introduction. Thank you so much for the time.
Thank you very much for that presentation. It's already started to stimulate some questions in my mind, and hopefully in the audience as well. Before we move on and I introduce our first panelist, I would just like to mention that we have closed captioning available for this forum. There's a CC box on the screen. You can click that and choose language, so please feel free to do that. And also just reminding everyone to please feel free to enter questions into the Ask a Question box, and we'll try our best to get to all of them during the Q&A session. So thank you again, Chris. Moving on to our panelists. Our first panelist is Dr. Christina Faust. She is an NERC Independent Research Fellow at the School of Biodiversity, One Health and Veterinary Medicine at the University of Glasgow. Her research focuses on the impact of anthropogenic change on pathogen transmission among multiple host species. Current research projects focus on how time since land conversion impacts disease dynamics among small mammals in eastern Uganda and impacts of reforestation on rodent communities and viruses they carry in Scotland. The ultimate aim of this research is to identify mechanisms driving transmission in a changing world and provide guidance on environmental solutions to reduce human disease risk. So please, Christina, if you don't mind taking the floor.
Great. Thanks, Ryan. And thanks very much, everyone, for the introduction, and especially Chris for kind of introducing some of what I'm going to talk about and kind of more generally. So as Ryan mentioned, I'm based at the University of Glasgow in the School of Biodiversity, One Health and Veterinary Medicine. And our team's research really kind of concentrates on understanding how land use change impacts host communities and the implications for zoonotic disease. So I'm going to give a really brief overview of some of the ideas. Some of this comes out from conservation ecology, and then the other panelists will also briefly touch on some other ideas. So there's kind of increasing evidence that land use change is linked to zoonotic emerging diseases, but potentially it's not always the rule. So we think that potentially that the highest disease risk, and that's interpreted as human cases of disease or kind of infection prevalence in vectors, is associated with areas where we have intermediate areas of habitat loss and potentially recent habitat loss. And there's a few disease systems represented here, but this is kind of a general thought that potentially when you have high amounts of hosts on the landscape, potentially they kind of can seed infections on that landscape. But this also suggests that these infections are transitory and potentially risk is not going to be consistent across space and time. So this idea that risk is not a static process is not a new idea. There's lots of kind of papers looking at this. This is some work that Andres Baeza and colleagues have done on malaria, and this is frontier malaria in South America mostly. And they've kind of done some really nice work showing that there's different phases of how malaria changes in response to deforestation. And identifying the perturbation that causes a change in infection dynamics, and then identifying when there's short-term and long-term outcomes that vary is quite important. So our group is kind of interested in identifying these phases of different emerging infectious diseases in order to be able to understand when and where we should be intervening in these systems. So fortunately, there's a lot of work that's been done by conservation ecologists. So this is a diagram thinking of, by a colleague that's done some really nice work looking at how changes in forest loss lead to changes in animal populations. And they collated a load of data from around the world and looked at how contemporary forest loss and time since forest loss affects populations of different vertebrates. And this is just an example of mammal species. And so on the x-axis here is the lifespan, and on the y-axis is the lag. So the lag is saying that after deforestation, it takes 20 years for a population that lives 25 years to stabilize. So there's lots of lags associated with these things. So even though we have deforestation, the host communities can take a while to change and adapt to that change. And because of that, my group kind of works on the left end of the scale. So we work on rodents and small mammals like shrews that live 2 and 3 years. So they respond really rapidly to environmental change. And they also are readily they're very good zoonotic reservoirs, and they also happily go in people's houses. And this is work we've been doing quite closely with colleagues at the Ministry of Health in Uganda. This is a map of Uganda in general. And more recently, we've been working on Buvuma Island in close collaboration with some local farmers. So Buvuma is quite an interesting island. This is a forest cover map from 2000. And in red is shown the forest that's been lost since 2000. This is just a bar graph of the amount of forest that's been lost every year for the last 24 years. So there's been a lot of deforestation on the island, and it's moved from secondary forest, pretty high cover canopy secondary forest, into smallholder agricultural fields. So we've selected a bunch of, we've recruited a bunch of farmers to participate in this study. And here's one of the farms here in Kekije. And so you can see there's still some trees on the landscape, but then there's also banana plantations, coffee, and some cassava in here. And what we're trying to understand is that when this deforestation happens, what is the change in the rodent community? How does that rodent community, the species diversity in that rodent community change? And how does the abundance of that community change? And how does that effect vary over time? So we use a lot of space for time experiments, as it's known. And we're using various bits of pathogen metrics in these populations, but one in particular that's quite prevalent is Capillary hepatica. It's a nematode parasite that humans can get, but it's also very promiscuous and infects a load of different small mammals. So here's some of our characters that we get in these plots. But it's quite an amazing amount of biodiversity that we see in these areas. In half a hectare, we can get up to 14 species of small mammals, and you can get some really interesting dynamics over time. So that study is ongoing, and we don't have all the results yet. And then just to kind of wrap up my little section. So we work on deforestation. And as Chris mentioned, reforestation has been suggested, and restoration of ecosystems has been suggested as an option for kind of preventing zoonotics. But actually, we know a lot about reforestation in terms of responses to biodiversity. We know it's not a mirror image of deforestation. So when you start to reforest an area, it's not a mirror image, and we're not just rewinding the tape. So how do these different ecosystems adapt and respond? And is there a chance that when we're reforesting, we might also be kind of introducing new disease onto the landscape? And that's not a reason not to kind of continue with these different outcomes, but it's important to understand the consequences of these. So L'Oreal, so fortunately we have some colleagues in Scotland that have set up this amazing network of woodlands. So there's over 100 woodlands that have been characterized, and in terms of their biodiversity, so tens of thousands of species from plants, invertebrates, and vertebrates have been recorded in these woodlands. And we know exactly how old each woodland is and how connected it is to other woodlands. And this kind of helps us inform future planning. So where would you plant, and how long would those forests take to reach biodiversity? So on the left here, we have a mature woodland, and on the right, we have a young woodland. And we've been doing some small mammal trapping with some PhD students and master's students. And we have much lower diversity in Scotland than we do in Uganda, but we do get a diversity of pathogens in them as well. And quite interestingly, we see that there's higher burdens and also higher diversity in young woodlands. This is kind of an ongoing research area, but we think potentially this is due to the microclimate that's in these woodlands. So these are quite isolated woodlands, and they are surrounded by agricultural areas. And in the young woodlands, we actually see higher temperature fluctuations. And because it's Scotland, it's reaching up warmer temperatures, but it's still not that warm. But these higher temperatures potentially are facilitating more transmission of helminth eggs and maturation of the eggs in the soil. But fortunately, we've gotten some new money from the European Union, and there's a whole suite of colleagues that are trying to look at restoration and impacts on infectious disease on a much larger scale. So just to give you a little sense of the spatial scale. So this is ongoing work where we're focusing on various viruses and bacterial pathogens in rodents and also bats in wetlands and woodlands. So hopefully there'll be some more data coming out soon. So as Chris mentioned, the empirical observations of these tend to be really difficult because you need lots of longitudinal surveys and spatially replicated, but hopefully we'll get there. And with that, I just wanted to give a brief overview and an opening. Hopefully other panelists will kind of talk about other areas, but I'd be happy to talk about exciting new technologies and kind of ways to take a whole ecosystem and whole pathogen approach to understanding these complex interactions. I just want to end by acknowledging all the funders and also amazing colleagues that have made this work possible. With that, I'll pass on to Ryan.
Thank you very much, Christina. Great to hear some of the earlier points reiterated in your talk, and I see plenty of questions coming through. So thank you to Christina, and thank you to the audience for your participation. Next up, the next panelist is Dr. Nicole Guntinker. She's a professor at the College of Veterinary Medicine, Center for Ecology and Infectious Diseases at the Odom School of Ecology at the University of Georgia, my home state. She is a disease ecologist with expertise in infectious disease ecology, community ecology, and wildlife ecology. Her research focuses on the ecology of multi-host pathogens and the impacts of anthropogenic environmental changes on infectious disease transmission. So thank you for joining us, and the mic is yours.
Okay, thank you very much. And it's an honor to be here among these great, amazing colleagues. Today I'm going to talk about something a little bit different, about structural dynamics that transcend ecological relations in relation to land use and infectious diseases. So ecological mechanisms by which anthropogenic land use and can influence zoonotic disease transmission, which has been touched upon by the previous talks, where environmental change could impact interactions between host vectors and pathogens by altering their population dynamics, that change their host or vector immune function or cause stress to them, lead to changes in vector hosts, the niche of vector hosts or pathogens, behavior, movement, or interactions of them. Changes in pathogen virulence or evolution are causing host switching, changes in diversity, community composition, structure, and traits of the vector host and pathogen, as well as altered food webs or trophic relationships. And we can see also that host vectors and landscapes are dynamic. So to the right, we see that populations fluctuate. Populations of hosts and vectors can fluctuate over time as well as over space in different areas. And as landscapes change, not all pathogens might have the same dynamic responses to these dynamic land use changes, such as deforestation and land reversion. They can vary. And land is under constant metamorphosis. So here we can see in this map that there are, in the areas of the red and orange, these are areas that have been under really constant, intensive, many years, thousands of years of intensive human use. And then in the purple, these are areas that have been in a bit more recently, in the last 200 years or so, under rapid land use change, due in part to what you can see that it's nearly the whole planet now. What Moore has called the Capitalocene, where these last 200 years have led to ecological transformation, discounting nature, and leading to the human footprint of biodiversity loss, habitat fragmentation, as we've seen here in the talks, disruption of food web structure and function, altered biogeochemical cycles, ecosystem regime shifts, facilitation of exotic invasions, pollution, and climate change. And so disease emergence can be seen as an expression of these ecological transformations. And when we look at them, so there have been many studies, and it's really important to look at these proximate mechanisms of how pathogen spillover occurs in response to land use change. And there are many different mechanisms of how zoonotic diseases are transmitted through water, airborne, soil, contact, indirect and directly with animals. And this was really beautifully illustrated by Plowright et al. And these are really, really important. These biological and ecological mechanisms of spillover are really important to get at. But stopping at this how this occurs and not the why can often lead to even blaming spillover victims. So often what happens a lot is that in some studies, there are common myths that emerge regarding land use change and disease emergence. One of them, to the right, there's a little drawing of this. To the right, we see the first one, which is the Malthusian myth, in that human population growth is outstripping. Once it outstrips resources, then we get disease emergence. Another one is basically blaming small-scale landholders for doing swidden agriculture and deforesting in these locations, or that particularly relevant now, that the myth that large-scale intensive agriculture is more beneficial to human health by their biosecurity measures and other things, as compared to biodiverse small scale or biodiverse agriculture or agroforestry. Another myth is that is often present is the that disease emergence is ultimately blamed on sustainable hunting practices or maybe small scale sale of game meat. And then the last one, where often we can predict where and when a spillover event occurs by feeding in a lot of data into models and understanding that this as a truth, that we'll be able to, by using AI, for instance, to just predict where the next large spillover event will occur and where the pandemic is going to come from, where the next pandemic is going to come from. So this is where structural One Health comes in. So structural One Health, a term coined by Rob Wallace and colleagues, formalizes the connections of the ultimate drivers of land use-induced disease emergence and spillover. It incorporates things like deep time history and pathogen evolution, cultural context, and circuits of capital and relational geography. And I'm going to focus on a couple of examples of this in the following slides. So here we see examples of land in motion. And this refers to the transition of land from use value, or land as a source of livelihood, to exchange value, which is land as a tradable commodity. And Bergman and Holmberg here in this paper, they mapped connections between land use economic activities, mapping commodity chains and capital in relation to land conversion. And they mapped 130 million direct connections with land use and economic activities. And you can see here in the result that where the arrows are going is where the economic benefit is going. And that you can see the driving forces for that land conversion, and the benefit is going to Europe primarily, and the US and North America. The drivers are not the landholders. Basically, the land use is contributing to growth and development of people outside the place where the land transformation is taking place. Therefore, this relational geography becomes paramount in understanding these processes. And then in relation to disease transmission, things that can happen when these land grabs take place is that smallholders are often displaced from their source of livelihood and nutrition, and you get increased food insecurity due to loss of land for subsistence crops and livestock, dependence on tenuous or temporary wage labor. People often get pushed to the borders at ecological margins where pathogen spillover often can take place. So you get increased human and domestic animal contact with competent wild animal reservoirs, vectors, and pathogens. Often people that are displaced to other areas are immunologically naive in their relocation site. And they can get moved to under-resourced urban or rural marginal areas with poor health infrastructure. And often all of these things can disrupt the social fabric also when people are forced to move from their land, or other members of their family are forced to go elsewhere to gain income. And this can cause intense psychological and physiological stress. And then all of these things together can increase vulnerability to infectious disease, zoonoses, and spillover. Another example would be Teachers Retirement Fund, the story of the Teachers Retirement Fund, TII-CREF, which is one of the world's leading, or at least in North America, retirement funds for teachers. I think my university also uses TII-CREF for their teachers' retirement plans. And a few years back, what they did was use they wanted to invest in land development in the Brazilian Amazon and other parts of Brazil. And so they, like sugarcane development and other large-scale soy development. And so what they did was use some indirect pathways for other Brazilian companies to bypass the normal legal channels that you would take for this, and invested a lot in land and in land development in Brazil. And so this then leads to the question of how was my retirement fund, or many people's retirement fund, could it have been related to the recent emergence or reemergence of yellow fever in Brazil, in areas of Brazil, which have been shown in many studies to be associated with fragmentation, deforestation, and often at these edges of land use fragments, there often are changes in the vector communities, and where they come in close contact with primates, come in close contact with people, and you can get spillover events happening. So in short, in a summary, basically disease ecology goes well beyond those ecological relationships between species. And land use cycles cannot be understood without understanding circuits of capital in modern day times. And disease emergence and spillover can be expressions of the capitalist mode of production. So maybe potential solutions could be working with communities on these disease issues, which many are doing here in this panel, and land repossession by the people, which requires political change, and as well as development of healthy agricultural matrices, things like agroforestry practices, and even incorporating technologies, but that promote a biodiverse agricultural matrix, and doing things like studying these agricultural matrices, as our colleague Christina is doing, are really important for understanding these risks. And with that, I'd just like to thank you all for your attention, and thanks. And thank my funders and my colleagues at the Gorgas Institute, where I do, I've done 15 years of work working on Chagas and Leishmania. Thank you.
Nicole, thank you very much for that talk. Again, we see some great questions coming in. Please keep them coming. Now, I would like to introduce our next panelist, Dr. Eric Lamlin. He splits his time between UC Louvain in Belgium and Stanford University, where he is the George and Setsuko Ishiyama Provost Professor and Senior Fellow at the Woods Institute for the Environment. He's a geographer and environmental scientist working at the interface of natural and social sciences. His research focuses on understanding the causes and impacts of land use changes globally and in critical environmental hotspots, with the purpose of informing sustainable land use governance. So thank you very much for joining us. Please take the floor, Eric.
Hello, thank you, Ryan. Hello, everyone. So I'm really looking at this from the land side of things. I'm what we call a land system scientist, and we're really looking at the human environment interactions on terrestrial ecosystems. And this field of expertise has been a long way from an almost exclusive focus on the impacts of land use change on climate, hydrology, biodiversity, to a much more refined understanding of what's driving land use change, looking at very long-term trajectories, looking at very fine-grained spatial patterns, a range of dimensions associated with land use change, as Nicole has just illustrated, but also all these cross-scale dynamics, these telecouplings. We initially, like 20, 30 years ago, we had this very simplistic, very rough view on this kind of transition from forest conversion, agriculture, intensification agriculture, urbanization. And now, we have a much more granular understanding of all these multifaceted patterns of land use change. This is just one example of a set of landscapes in northwestern Thailand, where basically you see this transition from the kind of landscapes with a lot of malaria, Anopheles mosquitoes, that slowly transform through clearing, irrigation, pipe irrigation, building little dams, tree plantation, all the way to urban, peri-urban, where suddenly now your landscape mostly dominated by the risk of dengue. One of our early work with my colleague Sophie van Wambeek really highlighted when we talk about land use, land cover change, there are really two complementary pathways. One is the land cover pathways, where the focus is on the disease ecology. So as we change land cover, the biophysical attributes of the land surface, we change the habitat for vectors and hosts, and therefore we change the level of hazard. But you also need the other pathway, which is the land use pathway, not just the purpose for which we are mobilizing all these biophysical attributes at the land surface, because these land use changes are really related to more human behavior and therefore to human-land interactions, and that influence the exposure. And it's really a combination of these two pathways that would increase the risk of land use change. And yet this representation is not fully satisfactory. It's too linear, just kind of one way from left to right. We really need to understand that more as a system, because of course, people, farmers, land users will adapt, modify their land management practices to mitigate or minimize risk of disease exposure. The same way as vectors and animal hosts of pathogens will adapt either their behavior or genetically to accommodate change in land use. And finally, people will adopt all sorts of prevention, protection, eradication strategies to decrease the risk of being infected. So it's really this kind of complex, multi-agent systems that we need to understand and represent. Just two studies to illustrate all this. This is a study with Laura Bloomfield, a former PhD student at Stanford, where she looked at the, she conducted some very detailed household surveys on farmers' spatial behavior with GPS. In addition, some very detailed, high-resolution remote sensing to look at the change in the spatial pattern of forest fragments. And basically what she showed is that the likelihood of contact between farmers and non-human primates increased significantly with first a land cover factor, which is the density of forest edges. The more forest edges you have, the more interaction you have between these non-human primates and these farmers, but also two key land use factors. It's the collection of small trees, mostly for construction by these farmers, and all the foraging, hunting for food into the forest. So you really have, again, this combination of a land cover change and a land use change. Of course, they are connected, but they lead to different types of interactions. In this case, it's really the loss of land cover, the loss of habitat that forces the non-human primates to interact more with humans. And as a small note, for vector-borne disease, in general, it's just the opposite. It's the habitats becoming more suitable for vectors and animal hosts that are due to land use change that increase the risk of transmission. Now, of course, today we have this very high resolution satellite data with very good time series, so we can really map at a very, very granular level this subtle change in landscapes. And we can also follow the human behavior, the spatial behavior of farmers, other land users, and really cross these kind of human behavior data with the remote sensing data, what we've always called kind of socializing the pixels, bringing social data in addition to these pixel data. And lastly, another study with my colleague Sophie van Wambeek. We really wanted to integrate a land system perspective in disease ecology. So far, the dominant focus has mostly been on the kind of direct, proximate effects of landscape patterns on disease risk. In this study, we really want to analyze how land use governance affects the risk of, in this case, tick-borne disease. We selected 3 case studies of tick-borne disease and really had a focus on the underlying indirect factors of land use change driving this risk of transmission. Of course, governance of land use shapes landscapes in ways that may be more or less favorable to ticks and to human-tick contacts. And this occurs mostly by structuring human-land interactions. And in these 3 cases that we reviewed, with the authors of this case being associated in that study, we identified 3 main factors that were kind of cross-cutting all 3 cases. One is the role of land use legacies, the kind of land use decisions over the very long term that keep influencing the way we interact with the animal host or vectors. Second, the various trade-offs that are underlying any land use decisions, trade-offs between conservation, production, aesthetic value of landscapes, etc. But also, importantly, social inequities, inequities in access to land resources, but also in access to the land management decisions. And this kind of ecological marginalization of some categories of some social groups is a key factor, and Nicole alluded to that already. So currently, land use governance does not consider its unintended effects, consequence on the risk of infectious diseases. And of course, a One Health approach does require mainstreaming health in all policies, including land use policies. So I suspect this might be an area of discussion in the Q&A that will follow. Thank you for your attention.
Thank you very much, Eric. Now moving on to our final panelist for this session, Dr. Paula Priest. She's a senior program coordinator for the Forest and Grassland Unit at the International Union for the Conservation of Nature. Her line of research focuses on trying to understand how to create healthy landscapes for people, with a focus on multifunctional landscapes with low risk of disease transmission, and while simultaneously mitigating climate change, providing ecosystem services, and also positive health outcomes. So Paula, please take the mic.
Thank you, Ryan. I'm just going to say that it's going to be really hard to follow all those amazing presentations, but I'll try, I'll do my best. I'm going to talk a little bit about how we are trying to plan for those healthy landscapes in tropical countries, and this is going to be based in a few studies that we are doing in Brazil, And just to give a note that I have a landscape ecology background, so the main focus of our research is trying to understand how we can really manage landscapes to make sure that they will have win-win benefits for both conservation and human health. And if you think about, we are always managing landscapes, like we are always deforesting, legally deforesting, and we are always restoring, even if indirectly. So what we're trying to do is really understand how we can, how, when, and in which context we can do all those management to make sure that we will have low transmission risk for people. And the first example, and it's going to be very fast because we don't have time, is about hantavirus. So we've been studying hantavirus in Brazil for the past 10 years, and I'm not entering any details here about the disease, but in Brazil, it is transmitted by these two very cute rodents. They are widespread, they are habitat generalists. And when we try to understand what landscape features really which landscape features are a driving factor to increase the abundance of those both species that are the main hosts of this virus in Brazil, we always see that is the amount of forest cover. So when we look at landscapes with different amounts of forest cover, so if you see this small landscape picture on the left, we have a landscape with 11% of forest cover, then 30%, and then more than 60%. And when we compare the abundance of those rodent species in those landscapes, we see that when we have less than 30% of forest cover, the abundance of those species can increase by 300 times. And when we test like several landscape features, it seems that for those both species, what is really important is only the amount of forest cover. They are not really being driven by amount of forest edges or other factors that can also be really important. So thinking about that, we have in Brazil a huge, so it is the country that has the highest deforestation rates of the world. So because of that, we also have a huge deficit in forest cover that we need to be restored to make sure that our legal requirements are in compliance. So we built this model where we were trying to understand what would happen with this region of Brazil, which is the Atlantic forest, if we restore the entire biome, just to make sure that our legal requirements are in compliance. So it's nothing that is absurd, but it just let's make sure that our law is being respected. And what this would do with the abundance of those two rodent species. And we're not really worried about how we would do this forest restoration, because for those species, what is really important is just increase the amount of forest cover. So we just create this model where we increase the amount of forest cover until our legal law is fulfilled. And then we saw that for both species, there is a huge decrease in the abundance in almost 50% of the entire biome, which is huge. So for one species, we see a decrease up to 44% in the abundance. And for the other one, a decrease in almost 98% in the abundance. So if you see, like before we restore the maximum abundance per pixel, that is the way we measure, it was almost 100 individuals. And after the restoration, it decreases to 10 individuals. And this is important because if we have a higher abundance of those rodents, they are going to have a higher contact rates between them, and the infection rates can be higher, and the chance of a person to be infected can also be higher. So thinking about that, because we do have this huge deficit in the amount of forest cover, and we have several restoration efforts in the region, what would happen if we restore those areas? And when we are restoring those areas, we have several ways in which we can do that. So we can increase the size of one large, big fragment, as in the image in the left, or we can increase the connectivity of this landscape, which is very good for biodiversity. So we can create stepping stones or just those small corridors connecting fragments. And for hantavirus, apparently, doesn't matter the way I do it. What is important is really just increase the amount of forest cover. However, when we start looking at other diseases, we see that it's so much more complex. And when we look at yellow fever virus, for example, we see that what is probably leading to increased risks of this virus is this high connectivity in the landscape. And in fact, we have some hypotheses that we believe that this increasing the amount of forest cover in the Atlantic forest region of Brazil could be one of the factors that led to the outbreaks that we just saw a few years ago that almost decimated our howler monkey population. We also saw that when you have a huge block of forest, as in the picture of the left, you can potentially barrier the virus movement. And this could be a potential ecological countermeasure to really avoid the dispersion and decrease the risks for this disease. So thinking about those two diseases, if I need to do a forest restoration or even legal deforestation, for example, the best way to do it would be, if I want to make sure that both hantavirus and yellow fever, they'll have low transmission risks, it would be doing in the way that I just increase the size of these forest fragments. And the problem is that there is no correct answer that fits all. When we start putting other diseases in this account, in this complex situation, we see that every disease will have this landscape signature, that's what we're calling, and it will have a different response. So it's going to be really hard to find a perfect, or like how I can really manage landscapes and make sure that I will have win-win benefits for all those different diseases, because they have different hosts, different vectors, they have different ecological requirements. And this, I'm only talking about the hazard. I'm not even talking about exposures or other factors that are really, really, really important when we talk about disease risk. And unfortunately, as everyone already mentioned in their talks, we almost don't know anything about restoration and the risks of those diseases. We do have a lot of research about land use change and the land degradation, but we don't really know what happens when we restore those forest areas. And this is really important because we are in the UN decade on restoration. We have this huge effort to restore at least 30% of all degraded ecosystems. And some estimates suggest that between 20 and 40% of the global land is degraded. So we need to restore a lot of area. And we have no idea of how we need to restore in which situation to make sure that we are going to have those win-win benefits for both conservation and human health. And this I'm only mentioning zoonotic diseases. When we start talking about other human health aspects, it gets more complex, or as mental health or fire-related diseases. So what we are trying to understand is in which situation forest restoration can have potential win-win benefits for people and can really decrease the risks of disease transmission for rodent-borne disease especially. So we have this project that we are calling ZooRest. We are trying to understand not only how land use forest loss, but also how forest gain is going to affect the community of small mammals, the community of predators, the abundance of those hosts, the virus that they carry, and how we can really plan to have those healthy landscapes for people. So what we are really trying to understand is what is the context, in which context forest restoration can have positive or negative effects for those diseases. And we have some hypotheses that they are all based on landscape ecology, thresholds, disease ecology, dilution effect, and we really believe that depending on the context, the way that you're going to do this forest restoration is going to be really important. So for example, if you have a low amount of forest cover in your landscape, you need to restore a lot of this landscape to make sure that you have a low risk in the future. However, if you have an intermediated amount of forest cover, around 30, 40%, the way that you do this forest restoration is going to be really important. In which spatial configuration you restore those areas is going to define those risks. And this is what we are trying to understand. So we have this very complex sampling design where we are sampling landscapes that have restored sites. We are controlling for the amount of forest cover in different situations. So we have landscapes with a low amount of forest cover, medium and high, and we are sampling in old forest fragments, new forest fragments that are connected to these old ones, and new forest fragments that are isolated from these old ones. And this is very preliminary because we're still analyzing the data, but what we saw so far is that we don't have a huge abundance of small mammals in these restored sites, so we are finding almost like on average 10 species of small mammals. And we just started the metagenomic analysis, but we found 11 virus family, 22 out of 700 samples. So I think we'll find a lot more. And we're also like trying to collect other animals as ticks and amphibians to see the like other mechanisms. But what we are seeing is that we had this hypothesis that potentially the abundance of those hosts, those rodents, would be higher when you have 10, 11% of forest cover, but we are seeing the opposite. The abundance of at least one species that is very important for hantavirus transmission is higher when we have around 30 to 40% of forest cover. So apparently what we're seeing is that when you have a low amount of forest cover, then the way you do this forest restoration is really important. And then if you restore your sites by creating new isolated small patches, then you can increase the abundance on those new isolated small patches. But if you have 40% of forest cover, it doesn't matter the way you do, you are potentially increasing the abundance of those species. What we are also seeing is that the context in which you do that, so higher the amount of forest cover around those restored sites, higher the richness of species. So we believe that potentially in the future, this could control the abundance of those species that can transmit diseases. And this, we are not even starting to look at this, but we also believe that there is a time lag for those ecosystem services of disease regulation to be returned to those landscapes. So it's going to take a while for those communities to be established again. And so we can see the abundance of those species to be controlled, and then the risks is really going down. We don't know how much time it takes. I know that Christina is doing an amazing job trying to also understand that. What we think for Brazil, at least, is that the forest degradation is going to be really important at this step. So now what we're trying to do is really understand what is the context when I do this forest restoration, but also what was the forest degradation stage when I start that, because potentially this could also be very important in defining those risks in the future. And I just want to acknowledge all the funders that fund this work, and thank you.
Wow, thank you very much, Paula, for that final panel presentation, and thank you to all of the panelists as well as our keynote speaker for these talks. Certainly got the wheels turning in my brain, and I see lots of questions coming in from the audience. And now I'd like to shift to the sort of the Q&A discussion session of today's forum. And you know, as I said, we have a lot of questions from the audience that I'd like to go through. Some have already been answered by the panelists during the talk, and I think sometimes it might be good to repeat those questions and give some of the other participants time to a chance to give their perspective on the topic. So I will repeat some questions that you've already very graciously answered in the chat box. I would like to start off maybe just with a little bit of moderator's privilege and ask a question that I have. We've talked so much. I think a common thread through everyone's presentation has been acknowledging that these landscapes are dynamic. They're changing, they're being degraded, they're being restored. There's certainly differences in disease risk and prevalence in different geographic regions, different climates, and associated with different land use. So I just would like to hear perhaps your individual perspectives, if you have one, on what would the ideal landscape look like. And I mean a landscape that is both home to us human beings. We are part of the ecosystem as well. And I think that's a topic that I think is important to remember, that we belong on this planet. We need a safe, cozy place to live. So what is this ideal landscape that is a comfortable, thriving home to us as humans, that is food producing, that both optimizes human comfort, food security, whilst simultaneously mitigating the risk of disease spillover?
I can start, but I've seen that everyone is so quiet. I think this is such a complex question, and I think it's really going to be context dependent. So the healthy landscape is going to be different if we're talking about the Amazon, if we're talking about the Atlantic forest, or if we're talking about Uganda potentially, or other region. What I think is interesting is that, at least like for what Christina was showing, we have very similar results between tropical forests and grasslands that we're studying in Europe. So I think probably we can find a general pattern. But I think is really context dependent. And for example, what we are seeing is for some vector-borne diseases, a landscape with more than 17% of forest cover is a high-risk landscape. But then when we talk about rodent-borne diseases, a 30% forest cover landscape is the high risk. And then when you have 70%, this is a low risk. So I think all those trade-offs, they are really, really complex. And how people are using those landscapes also something that is really going to define what is risk. So we have so many layers in this, involving those mechanisms, that I think we are just starting to understand a few of them. But I think is really context dependent, and I think is going to vary. And I think what is important is really to understand in which landscape we can increase those risks, because then we can really try to avoid exposures, because we have several ways that we can avoid exposure and try to work with behavior change and all of that. So I think if we understand the basis, we can do a very good job in really decreasing the risks.
Thank you, Paula. That was an unnecessarily complex question. If anyone has any other thoughts before we move on to the next one, please jump in.
Yeah, I think Paula's right in that there's not an idealized landscape. I think kind of strengthening healthcare systems is going to be important across the board, and in that, like, there's different risks for different places, and the better we can provide access to healthcare, so that if people do get sick, they have access to kind of rapid response is really important. And yeah, I guess that's kind of all I wanted to say. But yeah, I don't think I have in my mind idealized landscape.
Yeah, I could maybe jump in as well. I got asked this question yesterday as part of like a elicitation type exercise with the group that I'm collaborating with, and I found it really, really difficult to answer. And I think in retrospect, I think the reason for that is that, landscapes are comprised, if they've got people in them, of people with very, very many different perspectives and very many needs. So it actually doesn't matter that much what my idealized landscape is. What matters is the sort of sum of everybody's and the trade-offs and the the synergies that people are able to negotiate amongst themselves to have a landscape that can sort of sustain them, hopefully indefinitely. It's a very difficult thing to do. I've even asked ChatGPT to draw me a diagram of what the idealized landscape would be. It's always very nice, but I think it would just be so different and so context dependent. The other thing I'd add is that although I'm really sort of a biodiversity kind of fan. I want to spend most of my time in as natural and remote places as I can, looking at, you know, all of the amazing stuff that nature has to offer. I'm also a bit of a sort of a disease and parasite kind of fan as well. I just love the whole process of like species attacking other species. So I'm really into that stuff, but I'd have to say that this may not be the most important characteristic of a landscape. So I think if that was one objective, then that's fine, but it would need to fit in, like minimizing disease risk needs to fit in with a bunch of other objectives that all of these different stakeholders have for that landscape. And I think that's where it becomes technically and scientifically, you know, very, very challenging.
Thank you all for entertaining my first question. Would like to move on to some of the questions that have been submitted. This is for Chris, our first speaker. So it's going back to the beginning. You mentioned diseasey landscapes, but mostly focused on the human perspective. Is the diseasey landscape diseasey for everyone, or does the type of landscape that is diseasey depend on what the focal species is? For example, what is a diseasey landscape for humans versus shrews versus foxes? And of course, Chris, after Chris, if anyone else would like to jump in as well, that would be welcome.
Yeah, I'm probably the least qualified of this group to answer that question, but I mean, for sure that most of these models that sort of look at a net increase or an overall increase of disease risk has a target sort of you know, host in mind, and often that's us. So when we're talking about, you know, dilution effect, a lot of that work came out of, you know, work looking at Lyme disease risk in people that was associated with changes in biodiversity, changes in communities within forests that were, you know, in various states of change. I think there is this kind of idea in, you know, say this landscape immunity type hypothesis that there's sort of like a bunch of mechanisms that are increasing overall risk of pathogen exchange between the hosts that continue to exist in those landscapes. So I think there probably is something to that, but I think at the moment it's very, you know, it hasn't really been quantified from a multiple, you know, end point perspective where people are looking at quantified burden in, say, people, but also quantified burden in, you know, mice or frogs or whatever it is. And I think, you know, that they require very, very different study, you know, types, different methodologies. And at the moment, I think we're sort of picking around the edges of what is an interconnected system. We have some information, but the vast majority of the workings of that system are sort of currently obscured.
Does anyone else have any comment on that last question? Right. Again, I'll move on to some other questions. These have been already answered in the chat, but I would like some further comment from the panelists. Is there a clear role for protected areas in reducing disease spillover events, or is the research still presenting an unclear picture on the value of these initiatives? I think specifically in the context of conservation areas.
I could just give one small element, but it really depends on the level of encroachment, human encroachment into these protected areas. And there's always some, you know, there's none that's, you know, that is fenced by a wall, and there will always be, you know, people entering these parks either with their livestock or for mining or logging, and that's where the risk, of course, is created. So yes, the boundaries of these protected areas may be high risk zones for sure.
Can I just complement, because I also think that the way those areas are managed are also important, but not only consider the human side, but also the potential degradation that those areas have. So in Brazil, for example, we have several protected areas that are not well managed because we don't have resources. So we have this huge amount of forests that are really degraded. So we cannot compare a protected area that is degraded because it's not well managed with a one that is very well managed. So I think there is a lot that we don't understand yet about those nuances in degradation, those small scale threats that we have inside those areas that we need to understand. So I think it's potentially yes, but as Eric said, that is going to be dependent on several factors too.
Thank you. Moving on to another question, and this is one that popped into my head. I think it was during Nicole's talk. I was thinking about science communication. Denise Colberly asks, as a science communications researcher, I appreciated the nod to community and working within communities. What observations, if any, have any of you made about effective strategies for communicating this science to the community, policymakers, or other stakeholders?
I can try to answer a little bit from our experience about communication to the public, or we often work in multidisciplinary. Didn't talk about this work, but work on Chagas and Leishmania, 2 vector-borne zoonoses associated with land use change in Panama. And we work in multidisciplinary teams. And often in the process of, we work with anthropologists, cultural and biological anthropologists, and often in the process of interviewing people, we get an idea of awareness about certain pathogens, and often try to, at the end of our studies, provide a bit of information about the pathogens for people who don't know about them, and the vectors and transmission. Yet often that's not their main concern, are the pathogens. Their main concern often are other issues, right? Financial issues. And often their main health concern, at least in places we've gone, isn't infection, but it can be in other areas. It's often diabetes, heart disease in the areas that we're living, or childhood illnesses that are basically not zoonotic, but things like that. So we do try to go back into communities. We're actually planning to go back from our last study and communicate our results in a manner that is understandable to people. There are some people who have elementary level education, some people who can't read, older people often. And so we try to communicate our results in a way that people can understand and interact with them. But often after we get results back, we'll send, for instance, when we study dog diseases or exposure of dogs to Chagas and Leishmania pathogens, we will send those results back with some health evaluations via WhatsApp to the people that we've worked with. But yeah, I think communication is critical. And I think involving people, communities in science and what they want to see is critical. Often, I think we go in with our research questions, scientists go in with their research questions. There's often a need for those, but maybe those aren't the questions the community wants answered, or people from different areas want answered. Maybe we need to ask people, how can we serve you with the knowledge that we have in statistics or whatever, ecology, statistics, or disease diagnostics? How can we serve you better? Or what do you want from us versus us coming in there, which is typically the case to develop our studies, to maintain our careers. So, I mean, that's a really great question. Thanks.
Thank you, Nicole. The next question, Hans York Long asks, could you please discuss surveillance systems and risk assessment for potential spillover of filoviruses in view of recent Zaire and Sudan Ebola virus and Marburg virus outbreaks, and outlining pragmatic spillover prevention strategies in these areas? if we don't have specific experience with this class of viruses, which I think may be the case for this panel, could we talk a little bit about maybe surveillance systems in general, and how technologies are applied at surveillance? And I think it was Christina mentioned new technologies and a whole pathogen approach to surveillance.
So that's a great question. So there is some research that people have done looking at filoviruses outbreaks. So one of the challenges with filoviruses is a lot of the reservoirs, so the wildlife host that has them is not well known, or it's not exactly known. So people have used kind of machine learning methods to kind of predict which species are kind of most correlated with outbreaks. There's also some really nice studies using a case control design where you have an outbreak of Ebola virus or some other filoviruses, and then you randomly select other landscapes in that same ecological niche and try to identify features that are correlated with that outbreak location. So people are working on prediction of like identifying hotspots to kind of increase surveillance in those areas. But I think, so we kind of have some ideas of what ecological factors might be correlated with these, but it is very dependent on the data that's coming into these models. And some of the areas where we have these outbreaks, we don't have great surveillance. So I think more strengthening of surveillance, as Chris was saying, is like the data that comes into these models is really important. But there are kind of ideas and risk maps of these happening. And I would also say is that prevention is amazing, and it would be great to do that. But I also think that kind of detection of outbreaks and stopping outbreaks before they get too big is another kind of mechanism for controlling these. So sometimes they get out of hand, and certain countries kind of have more capacity to deal with outbreaks than others. And yeah, there's been several Marburg and Ebola outbreaks this year alone, and they've been handled in different ways. But yeah, it's all very challenging. But I think we have movement of where we think these things are happening. I don't know if anyone else wants to add.
Well, maybe quickly, there's in some circles it's being discussed, how to use some specific land use conditions as a sentinel for the risk of disease emergence. So then you can monitor, areas where these conditions are present. For example, areas with human encroachment into natural habitats, places with a lot of interactions between multiple species, especially wild and domestic species, transition areas between different habitats. So, I think we can do some special targeting to identify just based on land use, so not even waiting for the pathogen itself to appear, but the place where the risk is higher and therefore that deserve more attention in that respect.
Yeah, and also to say that the satellite imageries that are coming on now, we've got 30 centimeter resolution forest cover loss. So that sort of data is available now, whereas it wasn't available 20 years ago.
Yeah, I might just add a couple of points to that as well. I mean, I think, as Christina said, it probably doesn't pay to put all your eggs in one basket. You kind of need a bit of a spreaded approach, and that sort of primary healthcare component is really, really important because it sort of generically will likely help control and limit the impacts of any infection. So that that's really an absolute fundamental that everywhere can and should try to have in place. The surveillance, I find, is a bit more of a thorny question because a lot of surveillance is, it's not randomized. It's often targeted towards areas where we know or suspect high burden of disease or presence of disease, and that's if you sum that over all of the different diseases for which we're doing surveillance, that creates an extremely patchy picture. And it's the models that when we try to sort of integrate that information and correlate it with all of the conditions of emergence, that it's that sort of accumulated bias in the direction of attention that can sometimes be what you're modeling, and that may be quite different to the actual underlying risk. But I think, as Eric says, there's a lot of mechanisms and a lot of sort of theory that sort of feeds into all of that stuff that probably can, reasonably and robustly justify certain sort of spatial prioritization or areas of elevated attention. I think provided that sort of blind spot of research bias and surveillance bias is somehow explicitly accounted for.
I think that's a great point that Chris brings up regarding surveillance. And another thing is it's often really hard to randomly sample, or sampling is often most times opportunistic, because it's difficult sometimes to work in areas up steep hills or in difficult weather or other conditions where you can't sample wildlife. So it can be really difficult. And another issue is that often our studies, even most of the ones that I've done basically are cross-sectional. So following those things across things like El Nino events or other type of longer-term weather events, because hosts often cycle. So you might go out one year and you won't catch anything. It can be really difficult to continue those things without sustained effort and funding, especially because it's expensive to do. But yeah.
I would just kind of like to add to that point. Someone asked a question that I think resonates with the last point that you just made, Nicole. This was asked to Christina, but I think your last point touched on it. Have you looked into more passive surveillance techniques such as eDNA collection and metagenomics to determine diversity? Perhaps this could be applied to biodiversity dynamics in general, or the presence and dynamics of disease agents.
Yeah, so I think, oh, sorry.
Go ahead,
Nicole.
Christina. No, I was going to just say, this is Christina's wheelhouse here. But what we have done in some ways is look at blood meal analysis. It's indirect to see what animals might be there or contacting vectors, but a lot of people are doing that, and it's really interesting seeing what comes out. But
yeah, so I think Paula presented some metagenomics. So for people on the call that are unfamiliar with this term, the idea is that rather than targeting specific pathogens or specific hosts, you're taking an approach where you're sequencing everything in the sample. So it's an agnostic, semi-unbiased approach. So you're kind of sequencing anything that might be in that sample. And it's because sequencing has gotten a lot cheaper, people are using it more and more. It still is expensive. So like $300 for running a single sample is probably so people get around this by kind of adding together samples from the same species in one location to try to kind of increase your detection of certain pathogens and things. But it's a really cool method, and we're finding lots and lots of viruses, a lot of viruses that we don't know that were on that landscape before, but interpreting what that means for human health is quite difficult, because if it's 95% similar to a human pathogen, does that mean that humans are at risk on that landscape and everything? So it's really exciting what these methods are able to do. And there's some options that there might be kind of semi-quantitative. So in addition to saying this virus is present, we also see a lot of this virus in a certain location, and that might give added value. But I think the science is just kind of coming up to help us understand this more generally. And eDNA, I haven't personally used eDNA. Chris, are you guys using it in the Gambia?
Not yet, we're not, no.
Okay.
I mean, we are, but not in my studies. Yeah.
But there are some really like during COVID, people used wastewater sampling, and that people have used that for diseases over time. It does take a lot of optimization, because you are kind of filtering through a lot of things that you don't want to detect to kind of optimize for certain pathogens. So there definitely is a lot of technical expertise that's needed for those methods, but it's a really exciting way forward. Yeah, so great question.
If I can just add that, I know that the Pasteur Institute in Cambodia, they were using eDNA to try to identify virus in air samples in bat caves. However, it was bat caves with a very high virus load. So I don't know if the situation can be the same when you are inside a forest, for example, that with rodents, for example, that the virus load is going to be so much lower. So I think it's also going to be dependent on context, but it's very encouraging to see people already using it.
Yeah, it actually reminds me, I think one of the first eDNA studies that I sort of looked at closely was a pretty constrained environment in a zoo where they tried to pluck air samples and then identify the species in the zoo. And it was really good. It detected a lot of them, but then it missed some incredibly obvious ones that you would think would be just full, the air would be just full of like elephant, eDNA, for example. And that's, a level above the sort of pathogen community. So you really got to wonder about, how do you validate those types of tests? But yeah, for sure, a really, really useful tool potentially.
So, sort of going back to the theme of land use and land change, there was a question that I think was answered by Chris, but I think that it might be nice to get some additional perspectives on it. It was Jules Scottness-Brown's question about what exactly is the definition of land change. I believe they're approaching this from a social science perspective, but and part of the question is also about timescales. Landscapes have been changing way longer than the scale of our observations of them in relation to many of the diseases that are out there. So how much do historic landscape changes influence current disease transmissions? For example, in the northeast of the United States has changed rather dramatically from farmland to forest in the last 100 years. Has anyone thought about how these historic changes affect disease dynamics?
I can take the first part of the question and leave the second part for the disease ecologists here. So land change is really a shorthand for two things: land cover change in the biophysical attributes of the surface, and land use change in the purpose for which we are deploying, manipulating these biophysical attributes. And of course, this can be measured and studied at all spatial and temporal scale, from centuries to hours to days, and from a few centimeters to a whole biome. So it's definitely a kind of that concept is not attached to a specific scale. It just every study can be conducted at a different scale. Now, clearly, this kind of long-term historical change did have an impact on the disease burden. In addition, in combination with many other change, demographic change, economic change in nutrition, but I leave that to my colleagues here.
I can start. I'm sure that Nicole is going to answer that too. But I just want to include on what Eric said, that I think we need to do a better job when we say that land use change is one of the main drivers of disease emergence, because land use change can be, on my landscape ecology view, like any landscape dynamic that is happening on that landscape. So it's going to be forest loss, native vegetation loss, but also replacement of pasture to soybean areas, for example. And all these can have different effects on the host and vectors. So I think we need to, we're studying that, we need to do a better job in trying to understand this better. And about those huge historical changes, I think we don't have enough data. Just now we are starting to have data that spans back to 30 years ago or 50 years ago, that we can really try to understand those dynamics and really try to understand how this is affecting the current situation that we are seeing today. So this is, I think, an open question. Probably it has a huge effect, but I don't know, even if it is too long, maybe it doesn't have an effect anymore. It had, and then now the changes in small time spans are the most important ones, but this is something that we need to understand better for sure.
Thank you, and thank you to everyone for taking on these big questions and breaking them apart. And giving these really elegant, itemized answers to these big questions. I really appreciate it, and I know the audience does as well. So speaking of data, handling the abundance of data, Amosa asks a really thoughtful question here, thanking for the insightful talks. I'd say that there is an overarching but less sophisticated point. The complexity of our questions has outgrown the methods. The data available is very granular. The differences in answers that all these studies encounter tells me that we are in a very new frontier. I'd say, do you think that analyzing real-world landscapes and real-world outbreaks together is a good idea? Or one or the other would be better to first map out these emerging concerns? Another big one. Sorry.
I can start, but please help me. I would say that those outbreaks, they are really hard to be studied like when they are happening. So it's not that like we normally don't have the resources or like the opportunity to be looking at them at that moment. So in my personal opinion, this is my personal opinion, I would say that we need to understand better those mechanisms linked to land use change, to try to predict when those outbreaks can happen. And then maybe if we are really understanding those mechanisms, we'll be able to understand when an outbreak may happen. And then we can try to follow, because we'll be better prepared, and then we can really try to study this outbreak at that moment, trying to understand the mechanisms that triggered that outbreak. I don't know if this was clear, but this is my personal opinion.
Thank you, Paola.
I can add to that then, because it was a science. But I think it's very important to understand these complex systems. It's important to approach them from different angles, different perspectives. And one perspective is the kind of land use change, as Paula just mentioned. Another one is more on the kind of the pathogenic cycle, and climate also has a great importance, species interaction. But it's really bring all this specialist perspective on the same system. But at one point, someone has to bring all the pieces together. And that's the project of the One Health, perspective. It's so hard to do because you need on one hand the very deep expertise, but then you need the breadth. That's what we call the T-shape approach. T is the depth, you go down, and then the top bar is the breadth. And it's hard to do. And some people are better at synthesis, and others better at the kind of zooming in into a specific issue. But talking to each other, as we do now in this webinar, is so important for that exact reason. Because we need this combination of perspectives.
Thank you, Eric. And this also resonates with another question by Racek. Asked pretty much exactly what you just answered about the One Health approach and the importance of integrating both human ecosystem as well as animal health into this under the umbrella of One Health, integrating multiple scientific perspectives into a cohesive analysis and path forward. So thank you for kind of pulling it together. I have a lot of questions that I've been fed, so I'm going to try to go quickly. Let's see. Here's a bit of a philosophical question that I think is really important. It was in the chat. I don't think it made it into the Q&A, but moderators would like your perspective. It's a broad question. How do we balance that most finance and decision making comes from the global north, to use a contentious term? That most of the decision making in finance comes from the global north, but the burden is in the global south. Would emerging infectious diseases be a priority of the global north, given that most of the tropical diseases are associated with other services? You have cholera and sanitation being issues in the global south, whereas diabetes and such are, more of an issue of the north. What is the impact of these emerging diseases and the actual burden of the global north, as well as the global south?
I can maybe offer a few thoughts on that. I'm actually very curious to hear what Eric's perspective on this would be. But at least from the work that we've done, trying to sort of connect demand to demand on one side to impacts on the other, the main kind of threads you see across the globe are direct trade connections between wealthy countries causing impacts in producing countries which are often developing or lower middle income countries or middle income countries. The impacts seem to be they seem to be sort of proportional to those trade links. So it doesn't matter really what sector you look at. It seems to me that carbon emissions or biodiversity impacts and presumably other things that are all correlated with this trade route and land use specific impacts linked to commodities that are being driven by demand in wealthy places. I'm sure they're all correlated. There's some response to that in the sense that there's a lot of work around trying to educate or influence consumers on the one hand by illustrating and highlighting the impacts on producers on the other. Think of like bird-friendly coffee or farmer-friendly coffee and those kinds of mechanisms to try to sort of balance up the incentives a little bit on either end. But in my mind, it's like quite shocking every time one of these big input-output type studies comes out. It's really, what is really amazing to me is that it's just so, that there's on the one hand, there's demand, but on the other, there's acceptance in the producing country. So there has to be some recognition in the country that accepts that trade deal, that there are impacts there that they would need to quantify in order to understand the complete picture of the impact of that trade decision. And I think where you have very unequal systems in terms of unequal demand and producers, from a demand side and production side, it's hard to know how we would control this type of mechanism driving ongoing land use change, ongoing impacts, because the disconnect is just so high, through the global trade system. But this is not my area of expertise, as you can tell. I'd be very curious to hear somebody else's perspective on that.
Maybe just 2 quick points. It looks like the way the question was formulated is a kind of pre-COVID question, because wherever the disease emerged, we've seen that the whole economy, global economy, is affected in a matter of days now. It used to be in a matter of years or decades in the Black Death time, but now it's so fast. But then on Chris' point, I think it's 100% right. There's just another dimension that land use change, of course, is a source of negative impacts in terms of the environment and a number of ecosystem services, but often is also associated with some economic activities that may or may not generate wealth, and that wealth may or may not be distributed, generally not equally, but it may benefit some. You gave the example of coffee producers or cacao producers. These farmers really base their livelihood on these activities. So for them, that's the trade-off also. I need to generate some income and feed my family short term. And then, of course, these activities are transforming the environment, generating a number of externalities, including on health and health risks. But from their perspective, if they are at the margin of poverty, these questions are very difficult to arbitrate.
Thank you very much for your perspective on that question. Just quickly, we're running out of time, and I noticed that there were 2 questions on the impacts of wildfire on landscape change and disease and pathogen vectors. One question centered on fires increasing the likelihood of yellow fever. Could we get your perspectives on the relationship between fires and landscapes and disease?
I can start. So what we know, at least in Brazil, is that when you have fires, you can potentially increase the abundance of those hosts that can transmit hantavirus after the fire, because the new vegetation that is going to grow is going to be so rich, is going to be a very good food resource for those rodents. So they're going to come back, and apparently they can bury themselves into the soil to protect from the fire. So it's not a huge problem for rodents. However, for yellow fever, you need to have the howler monkey or other monkey species there. And then I'm going to say it's going to be dependent probably of the amount of fire and the context of this fire. Because if you really also kill not only the howler monkey, it is the most important one, but all the other monkey species, potentially, even if the mosquitoes can survive, you won't have yellow fever happening, at least in Brazil. So I think it's really dependent on the amount of fire and the context in which this fire is happening.
And to follow up on Paula's point, and in other contexts, fire could reduce disease transmission. I know in the Southeast, there's often prescribed burns to manage forests. And in these prescribed burn areas, it often reduces tick burdens on animals for at least the short term until the brush grows back up again. So really that context, like you were saying, Paula, is really important to look at.
Just to add one observation from the Gambia where we are, we don't yet have the data to really link fire patterns to disease, particularly in people, or even necessarily what's happening with hosts and vectors in the landscape. But it is one of the major things that prevents effective restoration that's trying to occur here on the ground. So some stats I've seen for some of the bigger restoration projects suggest that the patches of forest that have been part of the restoration activities are on average burnt every year, which is unbelievable when you consider that these areas are, there's replanting occurring. A fire will come through and completely wipe out the full manual effort of trying to restore through, for example, regeneration. So it's a very large barrier to active restoration in some landscapes. That said, there's lots of perspectives about the value of restored landscapes or fully or healthy landscapes in preventing fire. So a lot of people have strong belief that well-developed forests that they're stewarding are fireproof and things like that. So there's this really interesting social sort of perspective and social feedback with landscapes that have everything to do with fire and fire management, and 100% it's affecting the ecology of disease in these systems.
Thank you all. Thank you all for very thoroughly answering that question and providing really good feedback. I think we just have time for one more question, and it's very, very broad, very, again, a philosophical question. I was really happy to see it come through. Jamie Reeser asks, they're currently writing a biodiversity chapter for the 4th edition of the textbook Environmental Health from Global to Local. If you could transmit one message to future generations of planetary health practitioners, what would it be?
Develop a system thinking. It's really important to be trained at perceiving all these complex hidden interactions that can cascade through this complex system. That would be my 2 cents.
Mine is we need to, I think, work a lot with education for the new generation so they understand those links and they acknowledge that it's complex and that our health is at the center of all those changes, because then they would know how to choose better when they are voting and make sure that they choose someone that can really understand the importance of this.
All right. Thank you. Thank you very much for that last question and for answering all of the questions. We've come to the end of today's forum. Sadly, we've run out of time. I'm sure we could stay all day asking and answering questions. Really like to give a heartfelt thanks to our speakers, Chris, Christina, Nicole, Eric, Paula. Thank you for your fantastic presentations and the fantastic discussions. We at Cell Press, we'd also like to thank the audience for tuning in today to learn more and discuss more on this important topic. We went through a lot really quickly, so if you missed something and would like to go back and check on things, this has been recorded, and the webinar will be available for you to listen to and view the questions and everything again. It'll be posted on cell.com, so you can check it out on our publisher's website. If you have any questions about this webinar or suggestions for future topics, we'd love to hear from you. You can send an email to cpwebinars@cell.com, and we'll be sure to get back to you. So again, thank you, everyone. It was a lovely forum.
Thanks a lot. Thanks for having us.
Thank you. Thanks for the great questions,
Thanks.
too.
Bye-bye.